Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stolyarev II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings and welcome to our U.S. Transhumanist Party Virtual Enlightenment Salon of Sunday, July 28th, 2024. We have a fascinating conversation in store for you about senolytics and both their potential and their limitations in treating the problem of biological aging, which unfortunately affects us all unless we do something about it. So our panel for today consists of two of our U.S. Transhumanist Party officers, our current Vice Chairman and Director of Visual Art, Artemon Garcia, our Director of Longevity Outreach, Ben Ballweg, and we are also joined by David Wood, who is a frequent guest on our Virtual Enlightenment Salons, founder of London Futurists and Executive Director of LEVF, the Longevity Escape Velocity Foundation. Our special guest for today is Dr. Abdelhadi Rebbaa, who received his PhD in biochemistry from the Université Claude Bernard in Lyon, France. And he held research and faculty positions at Northwestern University, the University of Pittsburgh, and Columbia University. Over the course of his career, his research encompassed target and drug discovery and development in the fields of cancer, stem cells, and aging. He served as a principal investigator on multiple grants, published over 40 manuscripts, and reviewed manuscripts for several high-impact scientific journals. In his role at Ichor Therapeutics from 2018 to 2021, he oversaw the discovery and preclinical testing of small molecule senolytics and biomarkers of aging. He then worked for SENS Research Foundation between January 2022 and January 2024, where his role was to conceive, design, and oversee research projects on senescence prevention, senolytics, and partial reprogramming. And now he works as a project management consultant in New Jersey. So thank you very much, Dr. Rebbaa, for joining us, and please tell us a bit more about your work as well as your thinking about senolytics.
Thank you so much for the introduction, Gennady, and thank you all for joining. As we have discussed during the introduction beginning, I have some difficulty sharing my presentation as I'm talking right now from my cell phone. Is it possible? Is there any way you can guide me to do that, or will just because I have it on my computer, but it does not connect.
If you have your presentation available to send me via email, I can display the slides once I get them.
Perfect. Yeah, I will send it to you right now. Yeah.
Yes. But in the meantime, while you do that, perhaps you could give us some basic ideas about senolytics for those of our viewers who don't know what they are, what they do, and perhaps the current state of advancement. Just some high-level commentary might help.
Yes, yeah, I will right now. I'll just forward to you the presentation, and while you are working on it, I will try to open it. I will give some information.
Yes, excellent. And I will say, as a bit of background for our audience, I met Dr. Hadi Rebbaa at a virtual event that the SENS Research Foundation hosted in early 2022.
Okay, so
yes. And we had a few conversations.
Okay.
I'm sorry? Yeah, so as I was saying, we had a few conversations about his work and his research that led me to issue this invitation. And I did receive your slides just now. I will get them set up shortly. But I thought that the work that Dr. Rebbaa had done at SENS Research Foundation was quite promising. We aren't going to go into the details of this work in particular, but his more general thoughts on senolytics, I think, are very intriguing. And I am about to open the slides here. But in the meantime, my layperson's understanding of senolytics is they are drugs that essentially selectively kill senescent cells, which are old cells that have been damaged as a result of cellular aging. And by killing those senescent cells selectively, senolytics allow the non-senescent cells, the healthy cells, to perform their function better and not be subject to as many of the toxic byproducts. So I am going to launch this slideshow here and do a screen share. Let's see.
Thank you, Kennedy. This is very helpful. Sorry for the technical issue here. Perfect. This is very good.
Yes. So please
Thank
begin, and I'll get the slide view set up
now?
as you speak. Yes.
I guess I will just go ahead. Okay, I will just go ahead, and please interrupt me anytime you feel. So thank you for your patience, and today I will be going over some of the new approaches that are being investigated for the discovery of therapies to treat aging. Especially, I will be focusing on senolytics and how they compare to other emerging technologies. So the plan is to touch briefly on senescence, its causes, and key characteristics. And Gennady have already give you a good introduction on senescence cells and how we can target them with senolytics. I will also go through mostly the third generation of senolytics. They're small molecules, mostly derived from cancer drugs. And then I will talk about another generation of senolytics, which are the prodrugs, and also immunosenolytics, which are mostly CAR T cells. And then after that, I will discuss the potential challenges and limitation of senolytics approach from the practical and conceptual point of views. And then I will go over some proposed alternatives. As Gennady mentioned, senescent cells are mostly described as cells who are damaged and they're not functioning very well, and basically they are a burden to the good functioning of the tissues and the organism as a whole. And these cells are generated because of the accumulation of damage. As you all know, throughout our life, we are exposed to a variety of damages, whether from the environment or from within us. This can be UV light, pollution, ionizing radiation, smoking, our own metabolism when it's dysfunctional, and inflammation. All those cause damage to our molecules and cause senescence of cells, and this can lead to chronic diseases and accelerated aging. Over time, senescent cells are believed to accumulate in the body and contribute to the functional decline. Although the evidence for the accumulation of senescent cells is not really convincing yet, but it is something that is believed, and it makes sense that probably they do accumulate in the body and they contribute to aging diseases. So the obvious hypothesis is to get rid of these senescent cells in the hope that this will help us extend the health span. Of course, if we want to kill selectively or eliminate selectively the senescent cells, we need to know their characteristics and also their vulnerabilities. And from this aspect, the beginning of the discovery of senescent cells or research on this type of cells was actually done in cancer. And they were mostly characterized by the fact that they do not proliferate. They just stop proliferating, and they become flat, and they secrete molecules that are mostly cytokines that cause inflammation or cause fibrosis. Later on, they were also discovered that they cannot eliminate damage. They are getting damaged, but they cannot remove the damaged molecule within them. And because of that, the proteins are modified, and they express cell cycle inhibitors, and they become non-functional. So one of the markers, the common markers of senescent cells that were discovered early on, is called senescence-associated beta-galactosidase. This enzyme basically just cleaves galactose from molecules in the lysosome. And somehow this assay, although it's not perfect, but is used commonly to identify senescent cells, at least in vitro. Also, some people use it in vivo, but that's the assay needs to be optimized to really distinguish between tissues. So the role of senescent cells, although they were described, people do not know really for a long time whether they really play a role in aging or not. And this paper here described the first demonstration that senescent cells do indeed participate in the aging process. And the way they do it is that because senescent cells, they express a cell cycle inhibitor genes called P16, they developed an elegant technique to express the promoter of this gene linked to a Swiss side gene, which is basically a caspase. So when cells become senescent, they express P16 promoter. Automatically, they express the caspase, and that can be can lead to the death of senescent cells. So by doing this, as the mice become older, the senescent cells that are produced in them, they are eliminated automatically. So the mice that are not treated with the caspase activator, they become old, but the one treated with the activator stay much younger than the non-treated one. And the survival also was improved significantly, demonstrating for the first time that actually senescent cells do play a role in aging. And this stimulated the discovery of chemicals that can be used to selectively kill senescent cells. The reason is that chemicals can be controlled, it's much cheaper to produce, and also the genetics approach cannot apply to human for the reasons that it's complicated and even sometimes not ethical. So there was a series of senolytics that have been discovered. Mostly they target some intracellular pathways that are important for senescent cells, like a survival pathway, BCL-XL, the P53 pathway. All those are really important for survival senescent cells. So the first generation of senolytics, they targeted these pathways. There was also some research going on molecules or drugs called xenomorphics. Those, they inhibit or antagonize the expression of cytokines and growth factors, or like TGF-beta, that contributes to inflammation or contribute to fibrosis. Although at that time, there was a preference for developing senolytics, because if we can kill senescent cells, why try to inhibit only the secretion of SASP while letting this cells survive? So there was a lot of interest in developing the senolytics, but at the same time, it was interesting, but at the same time, it seems that it was going a little bit too quick. So in the beginning, they generated a lot of interest, of course, in the media and the press. And as you can see here, the MIT Technology Review in 2020, talking about the breakthrough technologies that most likely impact our lives. And the senolytics was one of them. And they even mentioned that actually drugs for senolytics may be available in the next 5 years from 2020. We are almost at that time right now. And we still have no drug, but it's generated a lot of enthusiasm and a lot of hope, but probably also a lot of hype. And some research went sometimes a little bit too fast. And one example of that is the drug developed by Unity, as you all know, that failed the clinical trial for osteoporosis of the knee. This not only affected the stock of this company tremendously, but actually it shattered the enthusiasm of many stakeholders in the senolytic field. So, but at the same time, it serves as a learning experience. That's something that we can learn from it, and we go to the board and try to figure out what went wrong, how we can improve it. So most of these early compounds that were discovered in the beginning, like dasatinib, quercetin, nuclein, etc., most of them, they have only modest senolytic activity, actually. And the one that we have tried many of them in our laboratories, and we could not replicate the senolytic activity by just comparing non-senescent cells and senescent cells and selective killing of these cells by these drugs. The one that works often for us and consistently is navitoclax, and worked for other laboratories as well. Navitoclax targets BCL2 pathway, which is anti-apoptotic pathway. And also it works very well in vitro, but in vivo it was problematic with this drug, because there are some cells that express also BCL2, and such as the platelets, and these cells also get killed by navitoclax, which can cause some side effects, thrombocytopenia and other diseases that can limit the clinical utility of this drug. So in order to go around that, a Spanish research group have taken this navitoclax and link it to galactose, a galactose that is substrate for senescence-associated beta-galactosidase. Luckily for them, when they link the galactose to navitoclax, that navitoclax become inactive. So that means even if you introduce it in cells that have BCL2, the target, it will not kill them unless these cells have senescence-associated beta-galactosidase, which are normally only senescent cells. So it was selective, at least in vitro, and we have actually tried it ourselves. It works very well in vitro. It does work almost like the parent drug navitoclax. It kills selectively senescent cells. However, when we can you please click the one more click? Yeah, no, back. Yeah, oh, it doesn't show it in there.
Yes, it doesn't show the chart, but I will try to produce it in another way. I just need to generate the image file for it.
Yeah, but I can just comment on it. When we did the chart, actually, the IC50, the curve was shifted to the left, almost like the parent drug. That means it does kill selectively senescent cells, but not non-senescent cells. However, when we conducted a pilot safety study in mice with this prodrug, the mice start losing weight and were not feeling well. So it was a no-go for us. We didn't pursue it further. Now we can please go to the next slide. And at that time, I was also looking for because I know that LEV Foundation were doing some research with this drug, and it seems, at least please let me know if I understood it, but it seems that it was not it was toxic also for this group of mice. If you see the two arrows, the gray and the yellow curves. So it looks like it's toxic, and when you think about it, if you go to the next slide, this prodrug target beta-galactosidase, and this enzyme is not only present in senescence. It's not present not only in senescent cells, but it's present on many cells, if not all cells. The only difference is that it's active at pH 6 in senescent cells, but not in the other cells. But this is only in vitro. In vivo, you can imagine if there is any variation in pH, some transient or sudden variation in pH, this enzyme will be active also in other cells, and therefore it may kill them, even though they are not senescent. The other thing is that regardless of the pH, it has been shown that macrophages also have beta-gal activity, although they are not senescent. So in vivo, this macrophage, or some of them, will also be killed by this type of prodrug, and that may explain the toxicity.
And we do have the chart now that you intended to show. So here it is. If you could summarize it once again for our audience.
Thank you. Yeah, what I want to say here is that it works very well in vitro. It does selectively kill senescent cells, the blue curve, almost as the parental drug, which is the pink one. And it does not kill senescent, non-senescent cells, the two dark curves. So it's really working very well in vitro. However, it was toxic for the mice that we are testing. So we decided not to go really with this drug.
Very interesting, and that's understandable that in vivo, since you've seen these toxic effects on the mice, that this would have dissuaded you from pursuing that drug further. All right, so now we have example 2.
Thank you. So sorry, I cannot hear you very well. It looks. Yeah, sorry, I cannot hear you very well. This looks like some connection things, but I hope you hear me.
Yes.
Do you hear me?
We can hear you. Yes.
Good. Very good. So so then this took us to investigate other things, other pathways, and with that, this is something I was working on it. I started working on it when I was in Ichor Therapeutics, now Ichor Life Science, is to target senescent cells based on their iron level. and to use a prodrug for that is activated only by iron. So the concept is, you have a drug in this schematic here is DR. It's called caged or protected by linked basically to a chemical molecule that protects it. So that drug is not active. But if it is exposed to iron, that iron will cleave that cage and release the drug. And the drug is toxic, so it can kill the cells in which it was released. So the concept here was used for cancer, because cancer cells are known to have more iron than non-cancer cells. But nobody at that time, and even until recently, probably were the first group to do that, to test it in senescent cells. And the reason is that a while ago, senescent cells were found to contain more iron than non-senescent cells. This is some early experiment in early 2000s by Bruce Ames, who did very elegant experiment. He took fibroblasts and continued to divide them for about 50 divisions, and the more they divide, the more they express iron on the panel on the right. And when they become senescent, they stop proliferating, but they continue accumulating more iron. If they take the same cells and may transform them, so they become almost like cancerous. So they continue proliferating, but their iron level do not increase. That means iron is actually associated with senescence. So it was very elegant experiment suggesting that iron can be a marker for senescence. And actually there are several studies that have shown that iron is increased in some with aging and in some disease associated with aging. So the only thing here is that they extract iron. They have to extract iron from the cells. You need to kill the cells and analyze iron by ICP-MS, mass spectroscopy. At that time I was wondering, is it possible to detect iron in live cells? Is there any probe or something that we can detect it? And if you go to the next slide, I use this probe here that was discovered just a year before we start this work by a Japanese group. They found this probe called S-ironox, and it's non-fluorescent, and when it's contact with labile iron, it's called ferrous iron, it becomes fluorescent. So we tested in our senescent cells, and as you can see in the right panel, all senescent cells that we have tested, they were positive for this probe, suggesting that indeed iron, again confirmed that iron does increase in senescent cells, and that actually we can detect it, of course, in live cells, and we may even detect it. We may even use it as a target to kill the cells. So in the next slide, a group led by Adam Renslow and UCSF, they made this structure that in which a drug, here's R, the drug R linked to NH, and they link it to a chemical molecule that is inspired from malaria drug, from artemisinin. And this scaffold have this endoperoxide that can be cleaved by iron. So when there is iron in a cell, it cleaves it, and the drug is released, and then it can kill the cells in which it was released. So in collaboration with my colleague Amit Sharma at SENS Research Foundation, we asked Adam Renslow to send us some of this prodrug, and we tested on senescent cells, and it works very well. It looks like it's working well. At least 4 or 5 times shift in the IC50, which is very good. But however, right now we don't know the toxicity of this molecule in mice, again, which is a key test. And also its toxicity and also the efficacy. We don't know if it will affect lifespan or not. So we are waiting for this experiment to provide some data. Also, this is a new field again, and one should be open actually to a surprising finding that may challenge our belief. Although this looks good, what actually this story is now taking a new turn and very interesting one, as we have found actually that a new target in senescent cells that is also increased in senescent cells, it's more potent in cleaving, in triggering this drug and activating this prodrug than iron. So this is something that we will be hopefully reporting on it sometime soon. Then last senolytic type I want to talk about is immunosenolytics. As you may all heard of them, those are CAR T cells. Basically, same thing. You need to have a marker on senescent cells. In this case, UPR, which is urokinase plasminogen activating receptor. This marker, although to my knowledge, I didn't see it often as a marker for senescent cells, but this group somehow they found that it is a marker, and so they have an antibody for it, for UPR. So what you can do in this case is just design a DNA sequence for only the portion of the antibody that recognize antigen, and link it to activating genes of T cells. So when these cells express this complex, they will recognize senescent cells, and then they will secrete perforin and granzyme and end up killing these senescent cells. This concept works very well for cancer, for certain type of cancer, and this group have applied it also to senescent cells, and it's a good breakthrough. So basically, to apply this to human is to basically take white blood cells and purify T cells, engineer them to express this construct, the CAR construct, and then amplify them, and then grow a lot of these engineered T cells, and then inject them back to the patient. In theory, this should find senescent cells, bind to them, and kill them, as I mentioned before. It sounds very straightforward. The group, Scott Law group, they use it in mice, and they found that it does reduce the fibrosis caused by high-fat diet in mice, causing liver fibrosis, but they could not see difference in aging, in effect on longevity, claiming because that the number of mice was not too high enough. The other problem, I think, probably with this technology is that T cells are the size are quite big, so they may not be able to infiltrate solid tissue. This technology was developed for hematopoietic cancers. It works very well for them, but it was not as good for solid tumors. So we can imagine that for tissues in the body or other tissues, normal tissues, it may work probably for some soft tissues, but not for solid ones. So that can be a challenge in using this technique for as a therapy for aging. Beside that, of course, it relies on a specific marker of senescent cell that is universal, and that's unfortunately there is none. as I mentioned, limited accessibility to solid tissue, that's another issue. But most importantly, this is the cost of producing these cells. They require a CGMP approach, laboratories and equipment, quality insurance, logistics and storage and transport. There is also strict regulation using engineered cells containing viral constructs and or produced by using animal-derived compounds. Also, the frequency of treatment and the cost and patient compliance that can be associated with that. And again, also the disparity of access to CAR T cells therapy because of the cost or because of all these problems, these issues. So although it's very encouraging and very interesting technology, I think translated into the mass, it will be a challenge. So now, if we just look at the senolytic approach in general, and is it good? Is it can be really helpful? There is, of course, concerns about the feasibility, as we mentioned, the markers, and also senescent cell heterogeneity. In every tissue, there is all type of senescent cells and all type of degree of senescence. And with that, the profile of senescent cells are different from one cell to its neighbor. And if one expresses the target of the drug, the other one do not. And so it will be a challenge just by looking at these markers and heterogeneity. The other concern, major concern, at least in my view, is the validity of the concept of targeting senescent cells themselves. Do senescent cells really accumulate with age? Although there is some indication, but the clear evidence, in my opinion, is not there yet. And how much can we quantify these cells? How many of them accumulate in the body with age? And is that enough to cause chronic diseases? And the other thing is, what about the beneficial effects of senescent cells? It has been shown for a long time that senescent cells actually can have beneficial effect, such as wound healing, they play a role in development. They actually facilitate regeneration. I remember in early 2000s, I was doing some training in stem cells, embryonic stem cells, and at that time we have to irradiate fibroblasts and make them senescent in order for stem cells to grow in them. So they support the growth of stem cells. So you can imagine if that's true in the tissue, it's really the play a role in maintaining stem cells. So assuming that senescent cells do accumulate with age and they are harmful, should we wait until they are produced to eliminate them, or try to prevent their production in the first place? As you know, from centuries and many civilizations, it says it's better to prevent than to treat. So that's something that I think we should think about it as researchers to come up with some other approaches. And if you just look at a cell from the time it is born to the time this cell is dead, of course it is born from stem cells. It can be young tissue specialized cells, YTSC, and it can mature to basically tissue specialized cells. Those are cells that work in that tissue due to the function that they were created for. If hepatocyte, they will do hepatocyte function. If they are melanocyte, they will melanocyte function. And they are healthy and they are doing exactly what they are supposed to do. These cells over time or by exposure to damage, they become old, almost like any live creature or human being. They become old, they become senescent, and from there they can degrade and die. So, and this can be just a normal differentiation development, or can be associated with stress and damage accumulation. Of course, here we talked about eliminating senescent cells using senolytics, but there are also other possibilities to prevent even senescent cells from happening in the first place. And one of them is to try to basically extend health span of a cell, of the functioning cell, of mature cell, and try to reduce the onset of senescence, basically by reducing the damage from reaching the threshold that is required for senescence. And we can do that maybe either by increasing the repair of damage in these cells or by preventing a damaging agent from accessing these cells. The other approach is partial reprogramming, which has been already started, and there's a lot of laboratories now interested in this. And this is also a promising approach that definitely can at least help cells become younger for a transient amount of time, and this way they can actually develop abilities to repair their own damage by themselves. But so reducing the damage is something that has not been investigated too much because there's too many damaging agents to deal with. But there are some that are within us. There are some that we can avoid sunlight, we can avoid pollution, we can try to avoid things. But some are within us. Some damaging agents are within us, and I think those we can at least focus on those and see if we can improve the outcome of aging. And by speaking about that, there's one potent damaging molecule within us that has been largely overlooked for its potential role in aging, is heme. As you know, heme is essential for life. It is contained in red blood cells, particularly in hemoglobin, and it serves a role in oxygen transport from lung to the rest of the body. It has a role in metabolism, or mitochondrial function, energy production, and activity of many enzymes. However, if not controlled, labil heme is extremely toxic and may precipitate the end of life. And there are some examples of that, like hemolytic disorders, such as like sickle cell anemia, and hemorrhagic crisis, like someone who have hemorrhage in the brain or any other organ. This the heme is so released in the system is so toxic that actually it can cause death. So we checked the level of heme in senescent cells versus proliferating cells, non-senescent, and we found that actually it increases in senescent cells. And we have developed a probe that can detect it in senescent cells versus non-senescent cells. And if you go please to the next slide, yeah. And if we give the cells, both cells, heme, non-senescent cells do not accumulate it. However, senescent cells accumulate. So we have a system to retain it, even if it's come from outside. So the other thing we found is that heme is more toxic. The next slide, if you can see the charts, is more toxic to cells than iron. If we give heme the same amount of heme to the cells, they will become senescent. And if we give them the same amount of iron, they do not. They continue proliferating. They stay healthy. And that suggests that heme is not only increased, can be a marker of senescent cells, but it can be actually a cause of senescence and can cause aging. And in the next slide, we have done the same thing in endothelial cells, and we found that same thing, it does inhibit proliferation and senescence in these cells, and induce cytokines of IL-1 alpha, which is highly pro-inflammatory molecule, and again, it induces beta-gal, suggesting that it does induce senescence. So the pending question in the next slide is, what is the major source of heme? Where it come from under normal and pathological condition? And what are the potential toxic effect of labile heme in the short term and long term? And how to prevent damage caused by labile heme? And can heme targeting be exploited to treat aging and associated diseases?
So I do have some of the charts that were omitted previously. So if you could perhaps walk us through these as I show them.
Yeah. So this one, we want to make sure that senescent cells do indeed contain more heme than non-senescent cells. And yes, we verify it, and it have a significant increase of heme in senescent cells versus non-senescent cells. This one is comparing the same amount in molarity between heme and iron, labile heme and labile iron, to see how they affect the proliferation of fibroblasts. And as you can see, heme inhibit proliferation at high concentration, but iron at the same concentration have no effect, or virtually no effect. The same thing for endothelial cells, because heme mostly is released in the vessels. So we use these cells as a model to see whether it can induce senescence in them. And we found, yes, it was indeed the case. So that means if we have more heme in our system, endothelial system, it's very likely that it will disrupt the function of endothelial cells and our vessels. Yeah.
All right, very good. And now we can proceed to senescent red blood cells as the major source of heme in the body.
Yes. So that's where most of heme come, labile heme come. It comes from senescent red blood cells. A while ago, I was working in a blood transfusion laboratory, and when they talk about senescence, they don't mean senescent cells as we talk about. They only senescence for them is just senescent red blood cells, and there are millions of molecules of hemoglobin in each of them, and each hemoglobin contains four heme, so it's a lot of heme molecules. And these red blood cells, they live for about 120 days. And there's 20 billions of them cleared every day from our body. So senescent red blood cells represent by far the largest fraction of senescent cells in our body. And their content, which is heme, if it is released in the system, it's much more toxic and dangerous than the cells released from other senescent cells. This is just a funny note that they are actually more safe if they are not dead than if they are dead by Senolytics, because then they will release this toxic heme. So under normal condition, there is, luckily for us, there is a control of these senescent red blood cells. They are eaten up by macrophages, but some of them escape, and they can just be can release their heme and of course iron in the blood, and they release hemoglobin, which is actually containing heme, and this hemoglobin can be is a tetramer and can go and become a dimer, and luckily for us, there are some molecules that can quench it and scavenge it. As one of them is haptoglobin, and if there is heme in the system, there's another molecule called hemopixin that can scavenge it. So those are protective for us. But they get overwhelmed when there is too much heme released. For example, in hemolytic conditions, let's say sickle cell disease, this patient didn't survive for a long time. The survival, their life expectancy is about 52 years. and they contain their heme level in the blood is higher than normal one. Another example of pathologic conditions that lead to excess heme in the system is IVH, intraventricular hemorrhagia, in a preterm born infant. So those who survive, they have a lot of heme in the system and in the beginning of their life, and they live with sequelae in their brain that can affect their physical and mental ability for the rest of their life. But also, there are other causes of hemolysis, whether related to diseases, sickle cell disease, beta thalassemia, malaria, sepsis, surgery, viruses like COVID, trauma. All those can induce a release of heme in the system. There are some medications. There's a list of them there. But the one that people take most of the time, like NSAID and penicillin, and there's environmental factors, whether the radiation, airborne particulate matter, and insecticide and pesticide, and those are people exposed to them most of the time. So throughout the life of the person, there is a possibility that there is hemolysis going on in the system. The other thing is that this paper here described as we get old, our macrophages that are responsible for clearing these red blood cells, they lose this ability, or at least they're reduced, and so therefore red blood cells they release heme in the system extravascularly, and the level of iron and heme also they increase in the system. Another way is that these red blood cells, regardless of the state of macrophage, in the circulation they just burst and release heme because of the extracellular matrix. So we know that heme is extremely toxic. Next slide, please. Next one. Yeah, heme is extremely toxic. It can by itself induce more hemolysis of red blood cells. It can go to normal cells and insert in the membrane and create lipid peroxidation, and oxidation can induce damage in molecules of the cells. If it affects immune cells, it can activate them and induce inflammation. If it goes to the plasma, it can bind to proteins like lipoproteins or albumin and oxidize them, and these can induce atherosclerosis and other vascular diseases. So it is very toxic, and for this reason, it needs to be scavenged or to prevent this toxicity. So for this purpose, we developed a probe that can detect heme almost 500 times more than iron, very specific for heme. And we use this fluorescent probe to screen a library for molecules or drugs that can scavenge heme, that can protect cells from heme. And we find some hits here, and we test them on cells. So if you go to a couple of slides, one more, yeah, this one. When we take control cells, they're fine. When we take these cells and give them heme, they start to die and get damaged. But if we give them these drugs and heme, they are protected. So this is very encouraging finding. And next step is to, of course, test it in a preclinical model to see if it can affect chronic diseases, or maybe vascular diseases, and also hopefully in the future can be going to clinical trial. So in conclusion, I presented that direct targeting of senescent cells can be challenging due to the heterogeneity of these cells within and between tissues. and the absence of universal actional targets. The prevention of senescence by reducing the level of damaging agent may represent a promising approach to attenuate the onset and progression of chronic diseases. Labile heme resulting from steady state or pathological hemolysis is a potent endogenous damaging agent, and therefore the development of approach to remove heme or control its level in the system may have application for treating aging-associated disease. And for this, I want to thank you for your patience and for listening to this talk.
Yes, thank you very much.
Thank you.
And we do have some of the charts that were previously omitted as well. So this is the identification of a selective probe for heme.
Yes, yes. So this is a probe that we just want to show how specific it is for heme. It's really strong in detecting heme in the blue than iron. You have to put almost 50 times or something more iron than heme to just start to see, to start to detect it. For heme, it's almost a very low amount of heme can detect it. Yeah.
Very good. And then this chart here, the HTS for small molecules that target heme.
Yes, yeah, this is just a screen for molecules that target heme, and we get a number of them, a number of candidates, and I showed you in the cells only 4 of them that are very protective. Yeah.
Yes,
thank you. So this kind of, yeah.
Thank you very much. And I wanted to ask a question of you before we go on to panel and audience questions. This is very interesting because you mentioned that there are environmental causes, for instance, exposure to ultraviolet radiation or exposure to pollution that could
sorry,
affect...
I cannot hear you. I cannot. Let me... Can you please repeat just that, because somehow it cuts when I... can you repeat?
Yeah. Yes. So you said there are environmental causes such as exposure to ultraviolet radiation and exposure to pollution that can affect the number of senescent cells in the body. And you also mentioned in one of your slides that essentially it's not entirely clear whether there's a direct correlation between senescent cells and aging itself. So you have a slide here that provides all of these sources: ionizing radiation, air pollution, UV light, inflammation, metabolism, and smoking. So I'm wondering, are these external sources more impactful in terms of how many senescent cells they bring about in the organism as compared to the process of biological aging? Or is their relative contribution not known yet?
That's a very, very good question. It's yeah. Yeah, the answer is I don't know exactly how much contribution they do, and if you look at it, some of them actually, you know, we can some people, some area have less sun, some area have less in the world, some have less pollution or less radioactivity. Some people do not smoke, some they smoke. So it's hard to so the level of senescence, you know, can be different, but still I didn't see, and to my knowledge, I didn't see a difference in aging that much between we don't hear like if you live in the north of the hemisphere you live longer or not as you live in Africa or something. So it's I don't see I cannot see how they contribute really to the aging. What I may speculate is probably the endogenous stressors, whether inflammation, things that cause inflammation, metabolism, And as I mentioned, heme, something toxic that's created from us. Probably because those, even wherever we go, they are with us all the time. So probably those are more correlative to aging than the others. Yeah.
Very interesting. Thank you. And now we have a question from John McPherson, who asks, might it be better to use Senotherapeutics to heal and repair senescent cells rather than killing them? What do you think of this idea?
Yeah, somehow it cuts. I need to do something because yes. I cannot hear you. It's like very fragmented.
Mm-hmm. But the question is on the screen, so I put up the text here. Can you read the question on the screen?
No, sorry. No.
Let's see. Let's try someone else, perhaps asking a question or reading out the question that's on the screen.
No, I cannot hear you. Sorry, I really apologize. I cannot hear anything.
Dr. Rebbaa, can you hear me? Testing, testing, testing. 1, 2, 3. Can you hear me?
Now I can hear you.
Yes, maybe perfect. It's just intermittent. The question John McPherson asks, a very good one. Might it not be better sometimes to use Senotherapeutics? No? Is it cutting out because of air conditioning?
Okay, now I hear you. I hear you a little bit better. Yeah.
So, can you read the screen? The question's on the screen.
Oh, okay. Might it be better to use Senotherapeutics to heal and repair senescent cells rather than killing them? There are xenomorphics. Xenomorphics are compounds that actually prevent senescent cells from being harmful. So these inhibit the secretion of these pro-inflammatory and pro-fibrotic molecules. So this way, the senescent cell will stay alive, but they are not harmful. I think this is one way to target senescent cells. The other one that have been started recently by David Sinclair is to reprogram senescent cells into a little bit younger type of cells. That's in vitro. Yeah, I think it needs some validation and some confirmation. But yeah, if it works, yeah, then we can actually heal them and make them younger and functional. Yeah.
Yes, thank you for that. And we have another question. Don't senescent cells need to be replaced as well? Like if you kill them, do you need something else to take their place in order for the body to function properly?
No, I think I don't think so. I think first there are only few. They're like 1% or less than 1% in any given tissue. Secondly, the space, they're very sparse. They're like one here and one there, one there. So if you remove one cell, that space that cells is taken in the tissue is like almost negligible. And I don't think, it needs really to be, it does not create a hole. It's not like something solid. It creates a hole. This is a flexible tissue and dynamic. I don't think it will create any hole. Yeah.
Okay, thank you. And Daniel Tweed wonders, maybe senolytic cells are displacing even worse biological actors. Do you agree with that possibility? Do you think that possible. Could be happening?
Yeah, it's possible. Why not? Yeah, they may actually push in, because they secrete these molecules that are actually important in communication, almost like neurons. Neurons, they communicate by secreting back and forth signals. So some are repulsive, some are attractive. So I may not be surprised that senescent cells are repulsive for some other cells that can maybe harmful to the tissue.
Yes, thank you. And David Wood asks, but the reprogramming of senescent cells to be younger is unlikely to be able to reverse genetic damage as opposed to epigenetic damage, correct?
True, but it reverses epigenetic damage. I think genetic damage, it happens when cells grow very fast, mostly like tumors, and because they accumulate things and can amplify them. The genetic, but genetic damage in somatic cells probably it does not amplify as much, and also there's a lot of mutation anyway in a cell, and a lot of them they're like benign. So unless that damage is affecting a key gene, in that case reprogramming will not help, because it cannot reverse the gene, the damage to DNA.
Yes, thank you for that answer. And now we have a question from Art Ramon. His question is there any benefit to frequent blood donation in lessening senescent blood or senescent cells in the blood?
I would think so. That if someone give a blood donation often, because at least in my view, from what I just explained here, that since heme can oxidize protein in the blood and can cause which can cause arthros and all these things. By replacing the blood frequently, it will remove these toxic proteins that contain toxic heme in them. So it will reduce the amount of these proteins. So it may be helpful for health.
Yes, thank you. And now another question comes from Ben Ballweg. He writes, you have been in academia, for-profit and non-profit sectors. What are the pros and cons of each, in your opinion?
The academia, I think they do solid research, but it's slow process. It's very slow and bureaucratic and all these things. The for-profit is there is less bureaucracy, at least in startup I work with and things. But sometimes they tend to go a little bit too fast without checking all the boxes, and it hits the wall quite often. The research,
Yes, thank you. And David Wood writes further, it seems that some kinds of cells cannot easily be replaced by multiplication of their neighbors. For example, neurons, rods and cones in eyes, and some cardiac muscle cells. So for these cells, killing senescent cells may result in holes that can never be filled. And our friend John H. also writes, this leaves the question of which is worse, the holes or the senescent cells. So what do you think about those cells in particular that can't be easily replaced? And what is the effect of killing senescent members of those cells?
Well, some people, it depends on the tissues. Some tissues actually they tend to get replaced, because as long as there are stem cells in that tissue, the stem cells will give rise to a replacement. And actually in every tissue, we don't have only stem cells and like mature cells and then senescent cells. We have everything in between, all type of cells in between. And when you remove some senescent cells, they're produced all the time. It's like you have, I don't know, in your garden you have tomatoes. Some they become red, you remove them, but then next few days you have another bunch of tomatoes, so you can go and collect them again. So they're produced all the time, because the other ones are like waiting in line to become mature. And the other ones are waiting in line to become senescent. So you remove one batch, and a few days later, they will be replaced. The question is, even a senolytic approach, if we treat a patient with senolytics, how long should we keep treating that patient every week, every month, every 5 years, or only one shot? And that's the problem, because they are producing all the time, even the frequency of treatment can depend on how much they are renewed. So that's one of the inconvenience of senolytic, is that we will probably never, they will always produce senescent cells and replacing the one that we removed. Yeah.
Very interesting. Thank you. And a related question from me is, are any senolytics currently approved or available for human use? That you're aware of?
To my knowledge, no. I didn't see anything yet, or maybe I missed something. But yeah, no, I didn't. I think the one that I'm aware of is the clinical trials that failed. But maybe some clinical trials are going on right now. But I don't know what's the result, and they're really working and validated by other clinical trials or by other groups. Yeah.
Yes, thank you. And another question from Ben Ballweg is, even though you're not aware of any senolytics that are currently available for human use, are there any health practices more generally that you would recommend based on your research that MDs might not yet be able to? And this could be as simple as, say, avoiding spending too much time in sunlight, avoiding air pollution to the extent that that's possible. But is there anything else that, based on your research, strikes you as particularly helpful for delaying senescence?
I would think that donating blood frequently, if you can, I think that probably will be helpful.
Yes, thank
because
you.
Because it's really, we are removing an endogenous damaging agent from our body, if possible, yeah.
Yes, and that's interesting. A bit counterintuitive, actually, from my standpoint, but I could see how that would work based on the research findings that you have outlined. Now, Daniel Tweed wonders, and this is kind of related to my previous questions, are there any senolytics that are available as nutraceuticals rather than pharmaceuticals that people might be able to purchase over the counter and would have senolytic effects?
Possible. I'm not, the literature, there's a lot of senolytics in literature right now. Some are from diet, some are from nutraceuticals. I admit that I don't really read all of them carefully to see what really, because the way we define senolytics in our research is really you have to kill senescent cells selectively compared to non-senescent cells. I don't know if this paper really did this kind of experiments to define them as senolytics. But in these compounds, maybe sometimes just xenomorphics that attenuates the harmful effect of senescent cells without killing them. It can be just anti-inflammatory molecules, because since senescent cells secrete pro-inflammatory molecules, that means if you inhibit inflammation, probably you are doing a xenomorphic effect. So I think the definition of senolytic, some papers they don't really respect it very well. So, but it's possible, yeah, that some nutraceuticals can also exert senolytic effect. I'm not aware of any one of them.
Yes, thank you. And an observation from Walter Crompton based on one of the slides that you showed. To his eyes, it appears that the SA beta gal results are essentially the same as controls. Would you agree or disagree with his observation?
Is it you say in one slide?
I thought it was one of your slides, but it may have been
possible. Yeah.
Yes.
Yeah.
Actually,
It's
I
possible.
think I can get to the slide in question. It may be slide 5 in your presentation that he referred to.
I'm not sure which one, but it's possible because it's a beta gallstinin itself. It's really can be very weak. As we mentioned, senescent cells do not represent a lot in cell population. Represent maybe 5 to 10%. This is in vivo. Even 10% is too high. But in vitro, we can get up to 90% of senescent cells. The only thing is that assay is really dependent on the investigator, on the experimental person. It depends on many things, and sometimes the scene is weak. It's hard to see the difference between control and senescent cells, and yeah, it's possible. Yeah, it's sometimes you really have to look at it with an ion expert to see. Oh, there is more senescent cell in this slide than the other one. But yeah, that's one of the difficulty of analyzing beta gal in general. Yeah, it's a lousy assay, and everybody recognizes it. That is not a good assay. But there is nothing around it. We have to use it because there's not many others. Yeah.
Yes, and I think clearly on your slide there's a difference. I think Walter was talking about more of his general experience with these results appearing as controls.
Yeah, he's right. Yeah, the most of the time, we have hard time proving even when generate senescent cells, we have hard time proving that they are indeed senescent. So in vivo is even worse. Yeah, much more difficult. Yeah.
Yes, and Walter, by the way, has given you a compliment here in our comments. He writes, I only know a little about Dr. Rebbaa's scientific work, but during my tenure as a volunteer at SRF, the SENS Research Foundation, he was always a bright spirit with a big happy greeting for me, whom he did not know at all. So some good words for you from our friend Walter.
Thank you, Walter. And Walter was an amazing person. It's very rare to see people like him who come to help without anything to in return or something. He just went to help an organization, and he was working with us and helping us with things that nobody will do, and he will do them. And I really my hat to Walter, and I have a lot of respect for you, Walter. Thank you.
Yes, indeed. And we are pleased to have Walter with us in the chat. Now we have another question from Art Ramon. Is young blood transfusion doing the same as blood donation in terms of reducing heme concentration? So if somebody were to undertake parabiosis, essentially, would that have a similar effect?
I would think so. Yeah, I would think so, because that young blood have not a lot of oxidized protein in it yet. So it's almost like taking the old blood and try to do some donation to reduce the amount of oxidized protein in the plasma. Yeah, I would think almost same effect.
Yes, indeed. And we also have an observation from David Wood, who writes that the rejuvenation roadmap published by lifespan.io currently lists around 20 different trials of treatments of senescent cells in human aging. Are you familiar with the rejuvenation roadmap and some of the trials discussed there?
I think I saw something on LinkedIn, regarding that. Yeah, I think I recall, yes.
Yes, very good. And I have always been impressed with the rejuvenation roadmap. I think it is one of the most comprehensive, let's say, overviews that are available about the state of progress in addressing and repairing the various types of aging-related damage. So I would encourage everyone to check that out. And we also have a question from Lillian Carlson, and This one is interesting. It wonders whether there might be any sort of difference in senescent cells based on whether somebody is depressed. So she is interested in finding out the difference in cells between people who are diagnosed and medicated for their depression versus those who aren't. Do you know of any studies that suggest that any mental health condition might affect the number of senescent cells in the body? Could there be that kind of impact?
Yeah, I would think so. Yeah, generally, depression is stress by itself. So, whether mental stress or physical stress, it will affect the well functioning of cell. I would guess, since it's depression, probably would affect mostly neurons or astrocytes or cells in the brain. It creates extra stress for them. So extra stress probably, if it's continuous and permanent, it may cause senescence.
It seems that a common theme here is the avoidance of stress as a possible strategy for reducing the accumulation of senescent cells. Very interesting.
That's correct. That's how I see it also, yes.
Yes, thank you. And there has also been a discussion of possible side effects of pharmaceuticals. So spatiality,
yes.
right, wouldn't any drug have side effects? And sometimes even the desired effect might be a side effect. She points out that the key is risk mitigation and the personal value that you have, and ideally the pros would outweigh the cons of particular drugs. And Lilium Carlson writes, it would be helpful to figure out which side effects are an actual indicator of harm versus a minor inconvenience in relation to longevity. And I wanted to hear more from you about possible side effects of senolytics. So we've got galactose conjugated navitoclax, which seems to not be particularly good for the mice. We see in the male mice, for instance, that those mice that have received the galactose conjugated navitoclax, they tend not to do as well as the mice that didn't receive it. And you pointed this out in your slides. So the yellow line, for instance, are the mice that received the galactose conjugated navitoclax. And you have the dark gray line, which is actually close to the red line for the mice that received all of the treatments. But the dark gray line shows the mice that didn't receive the galactose conjugated navitoclax. So what do you think might be happening here in terms of side effects? And would you expect similar side effects in humans?
Sorry, I start losing you now. Oh, sorry, go ahead.
Yeah, but
I just start losing you now right now. So, but I understand your. Yeah, I understand your point. I think definitely. I think one of the questions I was asking since the first few days I joined Ichor Therapeutics to work on senolytics is how to know a molecule is senolytic. Not only because it kills senescent cells more selectively than non-senescent cells, but that molecule should not be too toxic. If it inhibits proliferation as another side effect, that senolytic itself will become senescence inducer. So probably that's what happened also with GAL, with navitoclax GAL. But any senolytic that people are discovering, they never do that. They never check whether their senolytic itself, if they give higher concentration, will inhibit proliferation. Because if it does, it's very likely that it will induce senescence by itself. So if you look at it, basically, a senolytic will probably kill some senescent cells, but then induce senescence in a large number of neighboring healthy cells. It's very possible. And that's something actually it should be a quality assurance for every paper that talk about senolytic. They need to check whether that senolytic itself do not induce senescence. It's very easy. Just give it to the cells, dose dependent, and see whether it is staying with Isabeta GAL.
Very interesting. So the senolytics themselves might possibly induce senescence. That's a paradoxical kind of outcome, but definitely one to watch out for.
You're right. You're right.
So another
because
question.
They're stressful molecules too. They have also side effects. Yeah, as you said.
Yeah. So anything you introduce might potentially be stressful to the body. Interesting. Do you think any other senolytic would be less toxic than galactose conjugated navitoclax for mice or for humans?
Yeah, I believe so. Yeah. The navitoclax, the galactose conjugated navitoclax, as we talked about, is I think the target is probably the wrong target, because beta galactosidase is existing in many tissues, in many cells, healthy cells. The only thing that triggers it is the pH. So if the patient drinks something that reduces the pH in some area of his or her body, that's enough to activate beta galactosidase. And if it happens during the time of treatment with navitoclax galactose, then that will kill those healthy cells.
Yes, thank you. And David Wood points out that the latest results from the robust mouse rejuvenation study published on Aubrey de Grey's Twitter account on June 29th appear to show that the mice receiving the galactose conjugated navitoclax treatments live no longer than the controls. And that's the case for both males and females. So I showed the May 7th chart for males, but with females through June 29th, it seems there's also no significant difference from the controls. So that's very interesting. Now, we have a question from Elaine Walker, and this is related to Lillian's previous question. Would high cortisol levels tend to be an indicator of more senescent cells, or could they lead to more senescent cells over time?
That's I don't know. I'm not aware of the relationship between cortisol and senescence. Probably I missed some paper in literature lately or something that talk about that. I'm sorry, I cannot answer this question.
I think she was asking this question because cortisol is a stress hormone, essentially, and you were talking about stress.
Oh, okay. So yeah, in that case, probably yes. Yeah, anything that as you mentioned that can be stressful or toxic, or some level of toxicity, yeah, that's very likely that can induce senescence, especially if it is exposed for a long time. If it is like only for 1 day or something, probably not. But if it's chronic exposure to that stress or that drug or whatever, it's possible that it can result in senescence in certain susceptible cells. Yeah.
So once again, for anybody watching this who wants to derive an actionable insight, reducing stress, particularly chronic stress in one's life, can help to delay the accumulation of senescent cells. So thank you for that. And we've got a question from David Wood. He wonders, could you give a forecast for how the field of senescence and studying senescence might change in the next 5 to 10 years?
It's hard to forecast anything because there may be some surprise that can prove us all wrong in terms of senolytics or any other approach. But the way I see it right now, I think the prevention, it was not, unfortunately, was not explored enough. Not only for aging, but for most of other diseases, even in cancer. I worked a lot in cancer. And we always wait until people have cancer, and then we try to give treatments. Of course, that's for many reasons it's a good approach, because for pharmaceutical companies, this allow them to sell drugs and things. Prevention is hard to do clinical trial for that. Pharmaceutical companies cannot live on prevention because it takes years, and how can you do that in patients people who do not have cancer yet? For aging, I think same thing, but I think it can be more beneficial for aging prevention, whether try to avoid stress or try to reduce the damage caused by stress, or try to remove damaging agent if you can. Of course, we cannot remove some that we live in them, but some I think we may be able to remove them, especially inside the one that are inside us, secreted by our cells, or just a harmful thing that I think we can try to remove them. But I would say I think there should be some interest for prevention in the next few years. I think that will be very helpful for research and progress toward finding some meaningful impact. Yeah.
Yes, thank you for that answer. And David Wood also elaborates, for example, looking to the future, is it possible that the field of anti-aging will have a different set of fundamental targets in mind rather than the current hallmarks of aging? Do you think there could be some significant change in what are currently considered the hallmarks of aging?
Yes, I think there will be. I believe so, and not only with just educated guessing, but also now with AI, I'm sure there will be something there that some profiles, and whether profile at the molecular level or profile behavioral level, that can be detected as a hallmark of aging, that can help us guide future research toward another type of therapies.
So another observation that David made is there are lots of different aspects of aging, as we just discussed, and therefore lots of different sorts of therapies can help in various ways. Do you foresee any therapies on the horizon to address any of the aspects of aging that might be, say, complementary to the use of senolytics?
In general, I think somehow there has to be a combination of therapies. It's hard for me to see that a single target will really solve the problem, or single approach. It has to be a combination of approaches. Yeah, you need prevention, you need senolytics, you need maybe some reprogramming, you need different approaches in combination to have some meaningful impact. The only thing is, if you're talking about drugs, a combination of drugs, every drug have its own toxicity at certain point. So if you combine two, you're adding more toxicity. If you combine 3 or 4 or more, then you're adding more toxicity, and that's the barrier actually that will be needs to be overcome when we are talking about combination. But in principle, combination should help a lot. Yeah.
Yes, thank you. And John McPherson has a question along these lines. What would a complete program look like for repairing whatever kinds of senescent cells we can, and a senolytic cocktail to remove the other kinds which can't be repaired? He thinks eventually we're going to have an organized multi-pronged therapeutic protocol covering it all. Can you envision something like that, and the balance between repair and elimination of senescent cells?
Yeah, I agree. I really agree. I think it has to be a combination of, because there will be always some senescent cells. We need to remove them because we know that they are harmful. But if we can prevent them from occurring, or at least reduce their onset, that will be also helpful. We may not be do 100% of each. It will be at certain points, a certain percentage of each intervention will be effective. So we need a combination for them to increase that percentage of effectiveness. Yeah. But yeah, I agree.
Yes, thank you. And Mike Lazine wonders, doesn't your body completely renew itself after a certain span of time? I think he's talking about, for instance, observations that every 7 years or so, the cells in one's body are different from the cells that were there before? Or are there cells that are there for a longer period of time?
Sorry, can you repeat? I think I misunderstood that.
So I think Mike was talking about cells in one's body renewing themselves, like some cells will die, new cells will take their place. And after a certain span of time, is it the case that the body is comprised of entirely different cells from the ones that were there before, or are there still cells that remain?
Yeah, I think in my view, I think two cells will stay probably for a long time. Stem cells, those probably stay probably for most of our life, because that's where anything that we remove, that they are removed, it's replaced from stem cells. The other cell that may stay, and a lot of them actually, I agree with the question with Mike, that some of them may not last for a long time. Maybe some last 5 months, maybe some last 1 year or 2 years, or whatever. But some that last a lot is neurons, for instance. This stay for a long time. Cardiomyocyte also stay for a long time, for years, maybe for the whole life. And but the one that stay, in my view, all the time is stem cells. But all the others in between, yeah, they can be renewed, and at a certain point, the body will be almost completely new, except the neurons and stem cells and maybe some cardiomyocytes. Yeah.
Yes, and David Wood points out many of the neurons in our brain have been in existence our whole life, and there is some evidence for neurogenesis, new neurons being created, but not as many as you start out with. So you can't expect new neuron creation to replace entirely the ones that you started out with.
True.
So David wonders, looking around the world, where do you see some of the most innovative research being done? Or is all the best research still happening on the east and west coasts of the USA?
Yeah, I would think so. I think yeah. I think from what I see, I think I see a lot of reprogramming. I think that's very good, and it's in the USA. I think that's a very promising approach. Senolytics probably started here also, so same thing. It is in this part of the globe. Yeah, I think the prevention, probably, I don't know if somebody's starting somewhere. There's always, prevention has always been. It's just I think the focus and the longevity, like we're doing in startup and things, they are not a lot in that area. But if you look at just a lot of nutraceuticals and antioxidants, and all those actually are in intervention. So yeah, the antioxidant, they tend to work after the oxidation has happened. So then why not remove the molecule that caused that oxidation in the first place? So, but yeah, those exist already. But yeah, going back to the question, I think most of the research that is cutting edge right now is in the east and west of the US. Yeah.
This is interesting too. I'm curious why you think that is, because there are many talented scientists throughout the world. Why do you think the centers of research still are predominantly located on the coasts of the United States? And could there be in the future other areas, other hubs of research activity that could arise?
Absolutely, I agree that there are smart people everywhere, and but the volume of research and data that we see and papers that we see in this field, I think is mostly coming from the US. But yeah, of course, very likely that other area of the world will catch up on, even surpass this production here. I'm thinking maybe in Europe or China or Japan. Very likely that they will do more, probably more than here. Yeah. But right now, I think, especially the companies and in industry and a lot of interest and investors also, a lot of money is poured in longevity, in my view, at least from what I know in the US than other places.
Yes, thank you very much. And we have a question from Mike Lazine. He wonders, what is the age cap for these technologies to stop working? Is there an age limit where they cease to work? And for instance, with Senolytics, if you take an individual who is already very senesced, like a very old mouse, or maybe a human who is 90 years old, would a Senolytic be as effective in that individual as it might be in, say, a younger individual, a middle-aged individual?
Yeah, this is a difficult question. Yeah, if we think about the fact that senescence cells increase with age, so an old person will have more senescence cells to remove than a young person. So the dosage which should be higher of senolytic. But then when you increase the dosage, then probably toxicity will also increase for that same person which is frail and cannot handle toxicity as much as the young one. So it can be difficult, So in that case, the same dose of senolytic may not work as well for an old person than a little bit younger one. But it's difficult to answer. Yeah, it's and besides the older person is more frail and have other conditions generally that can maybe contract the effect or diminish the effect of the therapy.
Yes, that makes sense. And also, Walter Crompton wonders, is it correct to say that survival of toxic senolytic cells, I think he means senescent cells, is usually an immunity failure? So I think his hypothesis is the immune system should kill a lot of these senescent cells. And if it doesn't, it means the immune system is impaired in some way. Would you agree with that?
I do. I think, the immune system become old also, and so they become less effective at removing senescent cells. But removal of senescent cells is not done only by the immune system. They just die by themselves, or they die by whatever they die of. So one part of one contributing factor for that is sometimes also the immune system. So from that angle, if the immune system become old and senescence itself, there will be less removal of senescent cells. Yeah.
Yes, thank you very much. And there was a discussion here between Alan Crowley and David Wood. The essence of Alan Crowley's comments here is that there are some complexities in extrapolating evidence from mice to people. So if you have a study in mice and it shows something, either the senolytic works or the senolytic is toxic and has side effects and doesn't work, that may or may not translate into humans. So do you have any ideas or principles about how to interpret mouse data with respect to applicability to humans? Is there a way to think about the mouse data when we know that it may or may not have a similar effect in humans if you do a human trial with a senolytic?
Yeah, I agree. I think like any other diseases, its translation from animals to human is a challenge, and because the biology is different, the expression profile of molecules and targets are different. So it is expected that in most cases they do not translate, but sometimes those things are conserved between species very well, and with some luck, they translate. It depends on the target. Some targets do not exist in mice and exist in human, and vice versa. So it's just, and even if the target exists, there's a lot of other interaction between the cell target and surrounding cells and all the secretome around them and all these things that may affect negatively or positively the effect of the drug in between the two species. So it's a lot of things at play, but that's why mostly it's just a guessing thing to go from mice to human and from animal to human in general. Yeah.
Yes, thank you very much. And we are coming up on the last 7 minutes of our salon, and I am going to try to provide some closing questions or comments from each of our panelists, followed by an opportunity for you to provide any final remarks. And our first comment comes from Ben Ballweg, who writes, "The headlines I've seen lately about senescent cells highlight their nuance, how they may be net negative, but how destroying them indiscriminately could prove problematic, if not fatal. It makes me think the senolytic research route may be too challenging and not with as imminent a promise as other research routes. How do you justify the senolytic route maintaining its share of the anti-aging research industry as a whole, given its complexity?" Any thoughts on this?
I agree. I really agree. It's in the beginning we thought it's. Can I just go ahead and find the our charger, because it's almost my phone will. But I will continue talking. Is that okay?
Yes, please.
Yeah, but yeah. Sorry. Let me see. So can you please repeat a little bit the question?
Yes.
I think I lost the thing. Go ahead.
Yes. So Ben is writing that because destroying senescent cells indiscriminately could prove to be problematic, it makes him think that the senolytic research route may be too challenging and not with as much of a promise as other research routes. So how would you justify it maintaining its share of the anti-aging research industry as a whole, given that this is a more complex route than perhaps previously anticipated?
It is. Yeah. In the beginning, we thought that it's easy because I come from cancer, and I thought that killing senescent cells should be much easier than killing cancer cells, because senescent cells they don't mutate. So I thought, well, that should be super easy. Cancer cells they mutate and become resistant to treatment, and so those are much tougher to kill. But senescent cells will be very easy. But no, they are complicated, and one reason is heterogeneity. And cancer cells, the same thing. They mutate and become heterogeneous. So these guys, they don't mutate that much, but they are heterogeneous by nature because of developmental aspect. There are some young, some are a little bit more adult, some are close to senescence, some are pre-senescence, some are 1 degree 1 of senescence, degree 2 of senescence, degree 3 of senescence. So, and from one degree to another, they don't express the same target at all. And that's what makes it complicated and challenging to kill all of them at once. Sometimes some senescence, some non-senescent cells, we call it in cancer, we call it EMT, epithelial mesenchymal transition. Those are cells just differentiated from one state to another in response to stress, and they secrete a profile that is very similar to SASP. So those are not senescence at all, but they secrete a profile very similar to SASP. So you can imagine that even if you kill senescent cells that secrete SASP, you think that's it, I'm done with SASP. But no, because some cells that are just differentiated undergoing epithelial mesenchymal transition, they secrete the same profile. That means SASP is still there. And that harmful effect that we attribute to senescent cells, it's still there too. So Senolytic will not take care of that. And the problem will stay the same. So it is challenging, yeah. But mostly because of the heterogeneity.
Yes, thank you for that answer. and we have about 2 minutes remaining. There's an interesting question from our friend John H., who wonders, maybe we shouldn't be looking at destroying senescent cells, but rather rejuvenating them. He wonders, could we breathe new life into them somehow?
I think that's a very good approach, and I think that can lead to something really revolutionary. Yeah, I agree.
Yes, thank you. And we've got also a comment from David Wood. He writes, I guess solving senolytics is a bit like solving cancer or solving fever. It turns out in all three cases that a single concept needs to be broken down into multiple separate cases, just like the SENS approach broke down the damage of aging into 7 different categories.
Absolutely. They summarize it very well. Thank you, David.
Excellent. So we have essentially about 1.5 minutes left in our salon. Any concluding words from you, Dr. Rebbaa? Anything else you want to tell our audience?
Well, first of all, I want to thank you so much. This is an amazing discussion that we have. This is by far the best discussion I have on research topic that I worked on so far in my life. So I thank you so much for organizing this. And I thank the audience for the amazing question, very pertinent question. It's exactly the question that needs to be answered in this field for it to move forward. So I'm grateful for this day and for meeting you all, and I hope I can have the opportunity to meet the audience and meet you in person.
Absolutely. And our friend Daniel Tweed, who's also our vice presidential candidate, writes, our civilization's research scientists like Hadi are heroes of continued evolution. And Daniel says his cells are applauding. So I think that's...
Thank you so much. Thank you. Thank you.
That's the best way we can conclude our salon. All I can say is to everyone here, live long and prosper.
Yeah.